Hello everyone, my name is Tawfeeq Shekh-Ahmad. I'm a senior lecturer at the School of Pharmacy of the Faculty of Medicine at the Hebrew University of Jerusalem. And today I'm going to talk to you about the anti-epileptogenic and anti-epileptic properties of OMAVELOXOLONE. OMAVELOXOLONE is not yet approved for use by any regulatory authority. Let's first begin with a brief background on the involvement of reactive oxygen species, or ROS, and oxidative stress in epilepsy. I became interested in the recent years in studying the mechanisms underlying the development of spontaneous seizures following epileptogenic event, such as head trauma, stroke, or prolonged seizure-like status epilepticus. It is known that following a precipitating injury, there are several processes that occur, and starting right after the injury and can evolve for a very long period of time, which could reach even months and years. And eventually, these mechanisms will lead to the emergence of spontaneous seizures or chronic epilepsy. Among these several processes, excessive generation of reactive oxygen species and oxidative stress are perhaps the most predominant. But how does ROS involved in seizures and seizure-induced cell death? Upon the activation of NMDA receptor and the entry of sodium and calcium, these can activate several enzymes like NOS, NADPH oxidase, or NOx, and then leading to the excessive generation of free radicals or reactive species, which can interact with DNA or other macromolecules, causing damage to DNA, enzyme inactivation, lipid peroxidation, which eventually will lead to the activation of apoptotic pathways. In addition, the calcium overload in the mitochondria will lead to the opening of MPTP, leading also for the activation of several apoptotic pathways. So we have been looking at different strategies for targeting ROS and oxidative stress in epilepsy. And obviously, the first thing would come to mind is the use of antioxidant therapies. Indeed, these therapies of ROS scavengers have been extensively studied, including in clinical trials, but they have also largely failed. And this is due to several factors, including the poor characterization of the major or main source of free radicals generation during epilepsy, as well as the unfavorable pharmacokinetic properties of these antioxidants, which did not allow sufficient accumulation in the brain tissue, in addition to the low antioxidant potency. So we have been looking at alternative strategies, and the first is to identify and inhibit the major source of ROS generation in epilepsy, and the second strategy is to upregulate the intrinsic antioxidant defenses via the activation of NRF2 pathway. Therefore, we hypothesize that a combination of both of these strategies of decreasing the ROS production as well as increasing the cell antioxidant defenses via NRF2 is the most effective mean at preventing seizure-induced neuronal death, as well as epileptogenesis and epilepsy comorbidities. NRF2 is a well-known and studied master regulator of antioxidant response effect. It is widely studied also in the field of cancer. NRF2 regulates over 200 detoxification and antioxidant genes. It is normally regulated by KEAP1. KEAP1 is responsible for the ubiquitination and further proteasomal degradation of NRF2. But upon oxidative stress, NRF2 is released from KEAP1, accumulate in the cytoplasm, translocate to nucleus, where it binds to the antioxidant response element and initiate the activation and transcription of hundreds of genes responsible for increasing the antioxidant defenses by ROS scavenging, inhibition of ROS, then synthesis of glutathione, as well as increasing the ATP production through the mitochondria. What are other ways to activate NRF2? By giving exogenous molecules like OMAVELOXOLONE, which can bind to the cysteine 151 in KEAP1, then again release NRF2 and initiate all the process that eventually will lead to the increased antioxidant defenses. OMAVELOXOLONE is currently in clinical development by Reata Pharmaceuticals for other conditions like Friedreich's ataxia and mitochondrial myopathy. For the other branch of our hypothesis, we have been looking at the major source of reactive oxygen species in epilepsy. Of course, mitochondria was considered for a decade as the main source of ROS, including in epileptiform activity. However, we and others have shown that this is not the case. When looked at the mitochondrial ROS, or the reactive oxygen species of mitochondrial origin in vitro seizure-like activity model, we did not notice any increase in the ROS generation over time. When we looked at other sources like NADPH oxidase, we can see in red, this is the rate of ROS generation in seizure-like activity model, in the low magnesium model. And when inhibiting the NADPH oxidase with different enzymes, you can see that at all time points, there was a significant decrease in the rate of ROS production, which indicate that this enzyme, NOx, is a major source of ROS during epileptiform activity. So we want to look at the neuroprotective effect of OMAVELOXOLONE first in vitro. We showed that it can maintain the mitochondrial function and indicating that it can increase the production of or synthesis of ATP and preserve the abiogenesis of mitochondria. We showed that it can also decrease the ROS production following the pre-incubation of cells with 200 nanomolar of OMAVELOXOLONE. We also show that OMAVELOXOLONE can restore the glutathione in culture pre-treated with OMAVELOXOLONE and exposed to zero magnesium for 2 hours. And therefore, this can neuroprotect cells and decrease the rate of neuronal death. We then asked whether the combination of both OMAVELOXOLONE and NOx inhibition using AEBSF may have a greater neuroprotective effect. And this was the case in the dead neuron assay, where both treatments together had a greater neuroprotective effect and it decreased significantly the rate of cell death in vitro. We then wanted to ask whether these therapies, either alone or in combination, can modify epileptogenesis. We used the temporal loop epilepsy model of cyanuric acid-induced status. We induced status for 2 hours, terminated by diazepam, and immediately after we treated animals either with vehicle, single or repeated doses of OMAVELOXOLONE, single or dose or repeated dose of ABSF, as well as combination of both. And for looking at whether these therapies can modify chronic epilepsy, we waited 10 to 12 weeks after status epilepticus at the chronic epilepsy phase, and then we treated the animals with the same treatment groups. We first looked at the glutathione levels as well as the ATP levels in both cortex and hippocampus. Interestingly, following the vehicle treatment in animals received cyanuric acid and were induced for status epilepticus for 2 hours, there was a slight increase in the glutathione levels in both cortex and hippocampus. However, this was further increased by the treatment of OMAVELOXOLONE in a dose-dependent manner in both regions, and particularly in the hippocampus. When looked at the ATP levels, we noticed that despite the increase or the slight increase in glutathione in the vehicle-treated animals, in the glutathione levels, there was a significant depletion in the ATP in both cortex and hippocampus, which was again restored by treatment with OMAVELOXOLONE, again in a dose-dependent manner. We also looked at the neuronal and astrocytic cell death in the different subfields of the hippocampus, 1 week and 15 weeks post-status, and we showed that treatment with OMAVELOXOLONE, either by single dose or 3 doses over 3 days, significantly decreased the neuronal cell death in the different subfields. We finally monitored the development of spontaneous seizures in these animals following 2 hours of status. Animals received either vehicle or OMAVELOXOLONE, and you can see in the vehicle-treated animals there was the progressive increase in the seizure frequency for over 15 weeks. And when treated animals with OMAVELOXOLONE, there was a significant or dramatic drop in the seizure frequency that lasted over the 15 weeks of our trial. When looked at the latest weeks in this study, we showed that there was over 90% reduction in the median seizure frequency in the animals received OMAVELOXOLONE. We finally wanted to look at whether the combination of both NRF2 activation and NOX inhibition in rat model of temporal loop epilepsy can suppress the development of epilepsy. We again treated the animals with the different treatment groups as seen here, and we can see in all treatment groups there was a dramatic decrease in the seizure frequency, particularly in the combination group. As you can see as well from the cumulative number of seizures, the rats that received the combination of both drugs had or showed a significant decrease in the cumulative number of seizures. In addition, and importantly, we noticed that only the combination therapy had an effect on the latency period, where it was increased from 10 days to about 30 days following status. And finally, we looked at the seizure freedom in these animals, and we showed that 70% of the animals that received the combination of both OMAVELOXOLONE and ABSF became seizure-free from week 8 on. We then looked at whether these combination therapy can modify chronic epilepsy. We monitored animals 10 to 12 weeks after status for 4 weeks as a baseline seizure activity. We then treated animals with either vehicle or combination of OMAVELOXOLONE and ABSF. As you can see, there was a significant decrease in the seizure frequency in the treated animals, also seen by a significant decrease in the cumulative number of seizures in the treated group. To conclude, we showed that activation of NRF2 with OMAVELOXOLONE inhibited the ROS production, mitochondrial depolarization, and cell death in vitro seizure-like activity model. We also showed that OMAVELOXOLONE increased glutathione and rescued ATP levels in both cortex and hippocampus following status epilepticus in rats, which also was shown to prevented the neuronal death following the status epilepticus. We also showed that antioxidant strategies using the activation of NRF2 by OMAVELOXOLONE and NOX inhibition in where the first 24 hours following the insult has the potential to prevent the consequences of status epilepticus, mortality, and development of epilepsy and perhaps epilepsy comorbidities. This concludes my talk, and would like to thank all the contributors to this work, especially the people from the UCL Institute of Neurology. Special thanks to Professor Matthew Walker. I would like also to thank Reata Pharmaceuticals for the development of the materials. And thank you all for this virtual listening. Thank you very much.
Thank you very much, Dr. Sheikh-Ahmad, for a very, very, very interesting presentation. That's my opinion. We have a couple questions from the audience. One is from Anastasia Ribalco, who says, also, thank you for your talk. And could you please tell us whether OMAVELOXOLONE has potential in the treatment of stroke as a neuroprotective drug?
Well, I believe as a secondary injury of stroke is the involvement of oxidative stress and excessive production of reactive oxygen species. Blocking or inhibiting or decreasing this excessive production of ROS could be beneficial also in stroke and for other neurodegenerative diseases. But obviously, this need to be checked, and we haven't looked at these kind of models. But as a potential treatment, NRF2 activators are widely used in the stroke field.
And do you know if Reata Pharmaceuticals has any plans?
No.
Okay, just
we can suggest them.
yeah, exactly. Okay, very good. Another question by Steve White, who says that, to be clear, the effects observed on epileptogenesis was a result of a single dose at 24 hours post-status, question mark.
No, it was a single dose for over 3 days.
Okay.
So three doses over 3 days.
Okay. At the end. Okay.
Immediately after terminating the status of following 2 hours of status, we terminated with diazepam, then followed with single administration, and then the 2 days followed another single administration of the drug.
Okay. Another question, any speculation you might have regarding the adverse effects of this compound?
Well, it's interesting. We ourselves observed a very dramatic drop in the weight of the rats treated with OMAVELOXOLONE. And interestingly, the company is trying to approve the drug for weight loss. So this is something that is very clear that the drug does. What could be the mechanism of this? We can't tell. I can't tell. But it is definitely something to notice that it is associated with a significant weight loss.
Are you implying that ROS production might be involved in obesity?
It might be.
Wow.
Well, I don't know whether these adverse effect is associated with the ROS production or probably with another mechanisms that OMAVELOXOLONE is involved in. But this is something that we have noticed in our animals, and interestingly, it was observed also in humans in the clinical trials as well.
Regarding what you just said, I was very interested in your work. I went back to one of your papers, and I found that in the study of neuronal death following status, they used 25, or you used 25 milligram per kilo and 50. And the paper says that 25 milligram per kilo was tolerated and 50 had adverse effects. And therefore, from there on, which is true, what we observed is that after that, you always use 25 milligram per kilo.
Right.
So my question is, I noticed also in vitro, you used one concentration, okay, of OMAVELOXOLONE, which is 20 nanomolar. And in vivo, you used one dose of 25 milligram per kilo in general. My question is there any relationship? Have you measured the exposure to the drug at 25 milligram per kilo? And do you know if that concentration relates at all to the 20 nanomolar that you use in vitro for neuron protection, ROS production, etc.?
This is actually an excellent question. We haven't looked ourselves at the concentrations of OMAVELOXOLONE, neither in blood nor in the brain, but we have some evidence supplied by the company that the drug does penetrate the BBB, and there are sufficient levels of the drug in the brain. We have this evidence, but we haven't looked at this ourselves. We don't know what concentrations of OMAVELOXOLONE following these doses that we have used. Again, neither in blood nor in the brain. But we have kind of some evidence that it is penetrating the BBB.
But, you know, it would help a lot because it looks like you're going to be navigating. I mean, the effects are so great that you really need to know how best to use the drug and the minimum dose and obviously, you know, the concentrations. So if you can get a grip on that, I know it's not your drug, so but if you continue to work on it, if you can get a grip on that, you might be able to navigate, you know, the effects you want.
In fact, this is exactly what we are starting in my new lab at the Hebrew University to look at the pharmacodynamic pharmacokinetic relationship of another NF2 activator in plasma and brain tissue in animals.
Perfect. Wow, That's great. Don't go away. I think we have moved already into the general discussion, but the general discussion involves you and involves Dr. Lowen and involves Dr. Walker. And I don't know whether Torbjörn wants to be part of that. And so it's open for questions. I'm going to continue with the questions I have from the audience. Steve White said that your findings, Dr. Sheikh-Ahmad, are very impressive. That's the comment. It's not a question. And,
these are also Matthew's results.
I don't know. Okay, so Steve White, please write back. Tell us for whom. No, and then I have a question from Tatiana Stepanian that says, can the drug be first choice drug for MERF and MELAS, MERRF and MELAS? I assume it's for your presentation.
For my presentation?
Or I
think that's when the question came up, yes.
Yeah. In terms of time, that's what it is. Yeah.
Can you repeat the question, please?
Can the drug be the first choice drug for MERRF and MELAS? I don't know what
I apologize. I don't know what is MERRF, neither MELAS.
Okay, let me ask my co-chair, Torbjörn, are you around?
Matthew, you can.
Yes, I'm around, but I know that Matthew can answer this. He knows what MELAS and MERF are.
I do. Yeah, so we don't know. Is the question? I mean, one of the theories about some of the neurodegeneration that occurs in mitochondrial disease is through reactive oxygen species, and so it would be sensible to look in mitochondrial disease as well. But these are studies that are way into the future. So the answer is I don't know, but obviously it's something that needs to be looked at, and I think these drugs do show potential for those particular indications.
Okay, great. I have another question that's for Dr. Lowen. Was a drug interaction study by Neurocrine Biosciences conducted in healthy volunteers or patients?
It was conducted in healthy subjects. All studies to date have been in healthy subjects.
Okay, great. Thank you very much. Matthias Kopp, he says that these are mitochondrial diseases, so thank you very much. Mike Rogoski says the above question was not given to Dr. Lowen. Can you give it to him now? We did that. Steve White, for Tawfeeq, the degree of neuroprotection and anti-epileptogenesis is impressive. That's just a compliment from Steve White, anonymous attendee.
Thank you.
Okay, great. So thank you everybody for those, and we have another few minutes for general discussion. If anybody wants to bring up anything else on the presentations of session 5.
René,
Yes.
if I may, there were a couple of questions to Matthew and his talk.
Oh, please.
One more challenging than the other, and it's a follow-up on control studies and the possibility to carry out controlled studies. And the first came from, I think, the one you answered came from Roger Porter, and here's a follow-up from Ilo Lepik, making things even more difficult. Are you planning to carry out blinded studies, and would that be possible?
So again, I think my answer is much the same, that we would ideally like to carry out blinded studies. This was just an initial study to look at tolerability and doses. But the challenge is really what you use as your control, and how you control the diet similarly between the control group and the treated group, because you end up having to use some other dietary oil rather than beta shot. As I said, the concerns are that there may be multiple effects of other oils. So at the moment, what we're planning initially is to look at some specific indications. So we certainly want to have a look at those people with GLUT1 deficiency, and we will be doing a study in those, but that will not be a blinded study. And then the other thing that we do plan to do in the future is to try and do a blinded study. But as I say, there are logistical problems, and quite how we overcome those is not being solved at the moment.
Okay. I do have a question from Professor Bieler. What can you tell us about the chemical structure of OMAVELOXOLONE? I think the structure was shown one time in the slides, right, Dr. Shekh-Ahmad? But
Indeed.
Can you answer that question?
This is a second generation of alanine molecule. I don't know what Professor Bieler want to know more about the structure, but this is like we can give a bit ahead about the mechanism of the OMAVELOXOLONE. It binds to the cysteine in KEAP1, which lead to the release of the NRF2 from KEAP1. This is just a nucleophile that binds to the cysteine, the enriched in the KEAP1, and release the NRF2 from the KEAP1. Apart from this,
follow-up question. It's important to know the structure because, and you know the mechanism, with the cysteine, etc. Because then, in a new mechanism like this that seems to be so successful, it would help to have a series of backup compounds. I know if this was done in some other companies, obviously, and to see, obviously you maintain the mechanism, but in case, in terms of safety, all the other PK considerations, it would help to have backup compounds.
Well, indeed, there are quite an area of the derivatives of produxylin methyl. This is the parent. I assume this is the parent drug of this family, and these are about 2 times or even more potent. This family is about 2 times to 400 times more potent than other NRF2 activator, and they are assumed to penetrate BBB. That's why they seem to be very specific to NRF2 as well, and very potent in terms of NRF2 activation.
Okay, great. I'm going to try to sneak in. Yes, anyone? No.
No, I was just going to say it's a triterpenoid structure. So triterpenes are found in all sorts of animals, plants, and fungi, and so forth. And there are literally hundreds of them. So there are multiple different ones that could be looked at. And the companies identified a raft of these that are effective in as antioxidant compounds.
There's Adam Hartman combines, the 2 topics. Is there any evidence of what happens to reactive oxygen species with C10 or C8?
No, so we haven't looked specifically at reactive oxygen species in C10 and C8. That's something that we're doing at the moment. And also looking at the effects on mitochondrial function. So in parallel, Simon Heels, who's a biochemist at UCL, has been looking at mitochondrial function and shown that C10 increases mitochondrial function and also results in mitochondrial biogenesis. And so now we're looking specifically as well in those people with mitochondrial disease. The Murph and Melas from a previous question.
Okay, and you have a suggestion to you, Dr. Walker, for blinded studies. Could you use saccharin in the diet in one group and sugar in the other?
We could try. Well, thank you for the suggestion.
Okay, great. I think we're a little bit past our time, and unless anyone has any pressing question, we have a break for, it was meant to be 23 minutes.
Dr. Levy,
Yeah.
this is Gordon Lowen. Sorry, there are two questions that were asked earlier in the session on mine that I can really quickly address. So one was about a ketogenic diet, and I don't know the excipients off the top of my head for the product, but we'll certainly, as we go forward into the clinical program, we'll ensure that there's an alternative for those individuals who are on ketogenic diets in terms of at least the vehicle it's administered with. And then there were a couple of questions regarding any other seizures that we are targeting for the compound. And certainly we're hopeful to get into focal onset seizures, and we'll be evaluating other potential targets as well.
While you're there, Mike Rogoski asked, do you have any other indications such as pain for your compound?
No, not at this moment, no.
Okay, great. I'm glad we got that in. Okay, thank you very much. So we're supposed to be back here at 17:58 to start at 18:00. Thank you very much. Thank you, everybody.